Good evening, everyone. Thank you, Manjul. And thank you very much indeed to Rajiv Modi for inviting me here today. I come from St. George's in London. Some of you know St. George's. It is the home of Edward Jenner. He was a medical student at St. George's, and I actually work in the Jenner wing of the hospital. And it is out of the Jenner wing that Helper-B Therapeutics was born, and Helper-B Therapeutics is now in partnership with Cadler Pharmaceuticals. So I know what innovation looks like, and I very much admire the innovation which has been done in Cadler Pharmaceuticals, not only in the antibiotic field, which is my field, but also, of course, in the vaccine field, cancer, and heart disease, and infectious diseases, Rizarine. As Manjul has said, there is a massive problem with antibiotic resistance, which is going up like this, and unfortunately, the discovery of new antibiotics is going down to such an extent that in intensive care units in some parts of the world, only one antibiotic is left for very seriously ill patients, which is colistin. So what can we do about this? Well, the traditional way is to go down to the bottom of the sea, for example, or up to a very high mountain, and take a sample of soil, find a new bacterium, and isolate an antibiotic from it. Now, 20 new classes were made, 20 new classes of antimicrobials were made between the 1940s and the 1970s. And there's not a single new class has been made against gram-negative organisms like E. coli and Klebsiella since then. And this is why we are in this terrible mess. What is going to happen? Well, unfortunately, the benefits of modern medicine will be eroded, that is, cancer therapies, implants, immunosuppressive transplants, and organ transplants and such like. They will be blunted, they may even in some cases come to an end. So what Cadila Pharmaceuticals and Helper-B Therapeutics are doing is something which is rather different. Think of an antibiotic. Think of that antibiotic as being, unfortunately, obsolete due to resistance. Think of another molecule, B, a breaker, which can be added to that old antibiotic and rejuvenate it. That is what we are doing. We are doing it in the topical area and in the systemic area. And we are going forward with clinical trials, and we hope to be able to bring a new series of antibiotic resistance breakers together to the market. And the advantage of this is, I think, substantial, because if you think of your antibiotic A and your breaker B, if one breaker can service several antibiotics, then the subsequent combinations will be cheaper to develop. That's the first advantage. The second advantage, of course, is in 60 years' time, when resistance has arisen to the combination of the old antibiotic and the breaker, potentially you could produce another breaker which could do the same thing, so an antibiotic breaker C, for example. And this is the only platform that I know of which has the potential to produce antibiotics into the future by rejuvenating the old ones. Thank you very much.